Cool. So this is really what we're doing today. I tried to start up a lecture and then I was like, we're not gonna have enough time for me to like feel comfortable with you all having like 10, 15 minutes or so. So um does the group wanna be ambitious and get us started? Nobody?
Oh, I thought you were doing the one that's on the board.
Oh, I'm asking. I have four of these ones right now. Wait.
Oh, wait.
Mummies.
Yeah. Send it over.
What's up?
You can go first.
Great. Which one's yours? Your biochem?
Yeah.
Cool.
Oh, what? Oh my gosh. Cool.
Cool. Thank you for volunteering. Do you want to be in charge of slides or do you want me to just? Cool.
You can turn them if you want to. You want
Great.
something to do. That's fun.
Cool. While they're presenting and we're having our Halloween themed day, I've got a giant bag of candy. I don't want much of this left. So please take some, pass it around. Y'all can get some when you're done. But thank you for kicking us off on our fun Halloween themed chemistry discussions today. So biochemistry, get us started.
Right, so we had a paper about biochemistry changes that happen after death and basically what they found by doing that. And I just wanted to go over some things like that kind of helped build the experiment itself. Um, so one thing they were looking at are estimating the PMI, which was like the amount of the time after death, right? It's one of the most sought after pieces of information associated with the death investigations. And the level of understanding that we have of it is mostly based on observation rather than more concrete methods. So basically, they were trying to find a more concrete method of identifying that time. They used blood pH as a big indicator because it's one of the most regulated systems in the body. There's a very small margin that our blood pH has to stay in between in order for us to stay alive, which I thought was really interesting. And basically, once you die, obviously respiration ends and stops. It's no longer maintained. So the level of certain metabolites in our bodies change. And so basically they picked 6 different metabolites that they felt like that would change pretty significantly to study to see what their levels reach after a certain period of time. Basically, the aim of the study was to identify potential chemical markers that may be used to estimate post-mortem interval. And they were looking specifically at the metabolite concentrations of pig and rat corpses, and then the tissue degradation of human and rat blood in a controlled setting. So they were using both like things in a little dish and just corpses as they would naturally decompose.
Yep. Um, so there's a few things for the methods, and um, there's also, so, sorry, they mentioned the ethical considerations, because of course they need to get permission for getting the blood samples and the animals for they need sorry, they need to follow the animal cruelty guidelines. For sampling, they took the samples from the corpses of the pigs and rats, but then they also took blood and stored it somewhere else, like she said, from from alive humans and then also the dead rats. And then sort of go into detail about what they actually did. They sedated and euthanized, I think it was 20 rats and 2 baby pigs. And then they put a basically like sort of a collection tube for blood straight into their hearts. And then they repeatedly took blood samples over the next, I think it was 96 hours. And then for the blood samples for the humans and the rat corpses from the rats that they sort of stored, they put them in the same place to, maintain humidity and temperature. And then this was the methods for how they actually measured each thing. Blood pH was measured using a microelectric pH meter. The lactate and formate were measured using a spectrophotometric method, so like light. color. Concentrations of hypoxidine and ammonia were measured using the enzymic assay kits. Uric acid was measured using a reflotron meter and uric acid strips. And then all the analytical methods were based on like established protocols and were validated by using blood or plasma, to which varying degrees of target metabolites have been added.
Okay, now graph time. So, overall, looking at pH, you can see that in vivo, which is like the corpses, and the in vitro is like in the test tubes where they're doing all these testing and things like that. For in vivo, you're starting at around 7.5 for the rat and pig, and then it's going down at kind of a steady rate over there. You can see that when they die, before they died, it was 7.5, and then 96 hours of the death, it went to 5.1. However, in vitro, rat and human blood like did not move down at all. They were like, no. So 7.4 to 7.1. The rapid decrease in the vivo they said could be explained by the accumulation of all those metabolites listed, like Dylan was saying, like the bicarbonate and CO2 and hydrogen ions and things like that. Next, please. And then lactate and formate. So lactate is like a byproduct when you break down glucose in anaerobic glycolysis. And then formate, it kind of aids in synthesis of nucleotides, and it's a big byproduct of gut fermentation. Not gonna act like I know how all that works, but just so you know, that is that. So in vivo, lactate actually rose a considerable amount, but it rose a lot, especially in the pigs. And then in vitro, oh no, I'm talking about the form. My lactate graph disappeared. Okay, well we can talk about formate. Is my lactate over there? No. Okay, I don't know where lactate went, but basically I'm gonna talk
Was
to
it
you about
added
it.
in the last 2 minutes?
What?
Was it added like right before this presentation?
Oh, maybe more,
What?
mayhaps.
No, hold on. Just
She's gone. I don't know where she went.
seeing if I can exit and then come back and no magic.
It's all good because I can like talk about
Okay.
it if yeah.
Yeah,
So
go for it.
basically it rose in the rats and then it rose in the pigs too. Okay, wait, sorry, rewind. Brr. So rats it rose and then it plateaued at 9 hours and then it kind of like dropped a little bit, but it was like significant by the stats standards. And then with pigs, the same thing happened, but it did not qualify for significance. And then in vitro, it was like a non-linear increase again. It was like it rose and it plateaued and then it kind of dipped. And that's kind of where it ended after the 96-hour period. And then for formate, which I was talking about earlier, it rose rapidly. And then for in vitro, it did not really do that at all because it was so low that the assay they were using to detect that like enzyme activity like legit could not pick it up. So that's cool. Next. Thank you. So NADH, after death, the content of NADH in the plasma rose really quickly and linearly, from around 1.25 millimoles to around 28.5 millimoles, 96 hours after death. It kind of had a similar thing to formate where it kind of dipped at a 32-hour period. This time it was pretty like strong of a dip, and then it increased again, which is interesting. And then in vitro, human blood and rat blood had higher concentrations than the in vivo, and then kind of followed the same tiny increase after the 96-hour period. And then for ammonia, it increased quickly after the first 9 hours, peaking at 2 millimoles, and then it decreased to the point where once again the assay could not detect the concentration. For pigs in vitro, it increased before death, and then it from like 0.26 to around 12 to 12.5, which is kind of a big increase. For in vivo, there were higher concentrations found overall, which is kind of attributed to the fact that proteins, they can release them into different tissues, but like when you're measuring those proteins in like a little tube, they don't really have anywhere to like go. So that's cool. And then next. Okay, so for hypoxidine, In vivo, it was a steady increase. This time we're in micromoles. It was then again followed by a little decrease to the levels of being undetected. For pigs, it rose like crazy style. It was 1.7 micromoles to 6.7, which is literally an 1,000-fold increase. I think those are O's, not zeros. That looks kind of weird, but yeah, it grows crazy style. And then in vitro, humans started at 1 micromole to 18 micromole after the 96-hour period. And then for rats, it was a little different. It remained like completely steady between like 0.8 to point. That's interesting. Yeah. Over the 96-hour period. I don't know why I put 0.8 there, but yeah. Okay, last one, I promise. Okay, uric acid. In vivo, it increased like a lot from like below the sensitivity limit of the assay, which is 119, to 4,000 micromoles. For pigs, they were like, no, I don't want to do that. They only rose to around 170. And then for in vitro, it increased from the sensitivity limit again to 450 to 500 after the 96-hour period. And then for humans, it was kind of steady and it kept kind of low from the 200 to 500 micromole rep range.
So for limitations, one limitation was that there was a lot of, like you were mentioning, there was the sensitivity limit. So there was sort of a lot of variability for those lower readings. And something that they mentioned in the paper was that it was sort of a little bit unclear as to why pigs and rats had a huge difference in the concentration of formic acid. And they were saying that future research might be needed because it could be related to the bacterial microflora in like both of them. And then there's the obvious one where they couldn't really do the in vivo for humans because it's very hard to get a human cadaver for this type of experiment. But they also mentioned that it might be that like getting actual human cadavers might be not that helpful because of just how different people are to each other. And then the final thing was just that there needs to be more information about like how the actual temperature and humidity of it being stored might affect these conditions that are being looked at.
So what I felt was like really interesting about this article is I think that we think of like mortuary or coronary science as a very like concrete thing that a lot of us don't really know anything about. I mean, I don't. So it's very interesting to me to see that like the actual microbiome of the rats and the pigs was changing the rate of putrefaction, you know, the way that they were decaying. And I guess, is that something that you guys were familiar with, or is that something that totally new? Yeah.
Totally new.
Totally new. And then I was wondering, like, if this rate of putrefaction is different for everyone, do you think we could determine trends of what we'd see in their metabolites based on certain like microflora types? So if they have specific concentrations in their biome, is that going to correlate to however fast they're decaying or what they see there?
Yeah.
Okay. And then I was wondering how might age and lifestyle affect the severity of metabolic changes post-mortem? So like, what are some factors that you guys think would change the way that someone's body is decaying?
Is this a question?
Yeah, sorry.
Okay, no, I just wasn't sure if
Yeah.
it was like you were gonna,
Yeah.
probably someone's diet. I was thinking because like, I think it was King Henry VIII when he got put in his coffin, it started leaking because of gout he had. So that's kind of what I immediately thought of.
Yeah.
I-
What'd you say?
How they died.
Yeah, how they died. I was also thinking like animals in terms of like trying to correlate them with human bodies. Animals in general, their diet is pretty standard, at least for the most part, whereas humans, we're getting so much different input as far as like processed foods and all of these other things that we consume that I was like, are is using pigs and rats, is that even, like they said, is that even a decent indicator of what we can guess in terms of like human decomposition rates and time? And then does this make you feel any more or less compelled to donate your body so that they can study it? A little. It kind of made me more compelled. I was like, okay, I see. Yeah. Yeah, I think we think of it as like something that is very built out. Where I guess that's not the case.
That's awesome. Thank you all so much. Okay, there was one thing that I hated about this, not that you did anything. Don't.
Okay.
Okay, so let's go. All right, we're gonna look at this graph first, and this graph is totally fine because this was just looking at like lactic acid changes and stuff like that. And here we've got two of these graphs, and that makes sense that we're gonna have these two graphs, but they have very different scales on them. And we know that they are very different scales when we look at them, because like this one, it's an order of magnitude smaller. So that's good. I like this. The one that pissed me off was this one for pH. Because when I first looked at this, I thought, oh wow, this is definitely a downward trend. That I clearly see this downward trend. And then you had mentioned that like this wasn't really statistically significant. So these two graphs should have the same labels for these axes because they are comparing similar things here. And so that we could actually see where does this one place in terms of this one. So it should just be like a tiny, barely decreasing trend over there. And so having consistent like axes for graphs like this is really important because this one just looks misleading when I like first look at it. Yeah.
Why do you think they like opted for differing them?
I think they were just used to changing it for all the other ones where it does make more sense and probably didn't think it through. Like, shame on reviewer two for not yelling at them over this. But it's starting to get to the point where like it looks intentionally misleading that there is a more significant trend that is happening on the bottom graph. And then like you were the one who said like this is not a significant decrease, right? Like this is within our normal range of pHs.
Yeah, it wasn't, they didn't say anything about like statistical significance,
Mhm.
but in the article, they like clearly stated that like it was a very slow decrease.
Yeah.
Yeah.
So there's no justification for like why you have two different axes labels for this. And that's just one of the things that like kind of made me frustrated with it. If you've got big actual like a seismic change in your two samples, that's totally fine. That makes sense. But not for something like the pH one. Otherwise, this was a really cool article to go through. Loved it. Great. Good job, y'all. Who wants to go next? Keep the bag of candy going around. Come here. Antonio, which one was yours? You were mummies? Cool. Bring your team on up.
Well, we're ready. That's the thing.
You're ready, except for Olivia's not here, unfortunately.
Oh,
Olivia's sick. Are you able to do Olivia's parts and come?
try.
You can try your best. I will fill in gaps for what I remember on this paper.
Okay, we're talking about mummy. Next slide. What do we know? Honestly, not that much. Because the Egyptians decided that we're not good enough to get a recipe, so they didn't really write that down for us. But luckily, we have some of these Roman and Greeks down here who did write some of the stuff down. And our main one here is in the middle, Herodotus. He got this little list of ingredients here. Problem is, he came in a little bit late when they were kind of not very good at making mummies anymore. And this is also, gum is not very specific. We don't know what that is. So, and this is a lot of data, but I'm mainly going to focus on these graphs down here because I can't really explain the numbers, but I can explain a pattern here, which is these three samples down here, our S sample, our T sample, and our V sample, all have a very similar parabolic kind of shape to their graphs. And then these two other ones, our W and our U, have a much more like sloped, skewed kind of shape. So
Do you remember what all the like, just like what is A to W? Like, can you give me a little preview? of, like, what are we looking at here?
So they're measuring the number of carbon atoms and the relative abundance of them, which is we're just trying to like
In
figure
what
out.
kind of things? Like,
what-
do you remember what compounds?
So these two were sampling beeswax. This one was like the waxy ester compounds in
Cool.
it.
They're actually separated based on kind of row.
Mm-hmm.
So A through M is fatty acids. And then N through R is beeswax, samples containing beeswax, hydrocarbons of odd carbon number and neutral fraction from samples containing beeswax, obviously. And S through W is wax esters and even so it's even versus odd. Yeah, that's,
Cool.
I wonder what that could.
So we're looking at a bunch of different oils and types of waxes that were probably used in mummification. And we're seeing how many carbon atoms are still intact from them. Cool.
So beeswax didn't come into prominence until around like 1100 BC, which is like that's kind of our golden era of mummies. These are our best preserved ones. And we'll see there's a change in like the structure. We have our S and our T ones and our B ones, I believe, which are our earlier ones closer to our better preserved mummies. Then we start going on and we start getting those more sloped kind of shaped waxy ester things. So we can tell that something changed. Either they ran out of something, they had to change what they were using, or they just decided they wanted to use something different, but something they decided to change what they were using, and that eventually led to worse and worse mummies.
What does it mean to have a worse and worse mummy in this case?
Not as well preserved, more like rotten out.
Cool.
Okay. So this is a much more comprehensive little graph here. This is just showing us something. Okay. This is showing us kind of what was found, what traces of different substances were found in different mummy samples. So everywhere where you see a number 1 is data collected from the study done by these researchers who wrote this paper. And then there was also data used from 3 other references. So the level of darkness on our scales represents the relative abundance. As you can see, it's based on the different mentioned elements of like embalming fluid, and then and it shows over time the difference in relative abundance of each different material. So we can see that the methods evolved over time, and the article speaks about what this could maybe imply, perhaps over time learning about the like different antimicrobial properties or the different like sealant properties of these different substances and how they may contribute to the preservation of the body. So there's no ubiquitous method. We don't have one recipe. Okay, so this graph is evil.
We're gonna get to make
This
this
is the
graph in
scariest thing about today.
a couple weeks.
So I'll try to explain what I understand of this to you. So this is okay. Let's start where it came from. This was sampled from the resin of the top cranium of a late period male adult mummy from the the mummification period of 664 to 404 BC. It's a long time. So at this point, hold on, give me a moment. At this point, we kind of found that beeswax has become the most like widely used substance for preservation, and it was hypothesized that that was because of the sealant properties of beeswax, and then also the accessibility of different materials. So what we can see here is beeswax could be perhaps indicated by these black circles or these black upside down triangles like filled. And this is just showing based on our standard what the abundance of this cranium resin is for each different type of chemical that we are sorting through using a total ion chromatogram. And so basically we see there's the most of this and also this, and these are related to beeswax. These are like compounds that are found in beeswax. And then we see that there's like a lower abundance of other like plant matter and oils and things of that sort. So this is just for one sample.
Can I just explain this graph a little bit more? Cool. Because we're going to get to do a version of this. We're going to use the GCMS to find the concentration of caffeine in a few weeks in our gray water samples, as well as a cup of coffee. And essentially, what we're doing is we are pushing a bunch of carbon particles through a giant filter. So we've got the retention time where things that are really small are going to move through that filter relatively quicker at a shorter retention time. Things that are really big and really heavy take a lot longer. So over here, we are just getting, these are all of our smaller carbon sources that we have. Then we get to some of our mediums and then some of our large ones. The chromatogram essentially is just taking like a colorimetric reading of them all as they pass through at the very end. So however much time it takes for them to pass through, you get a different peak associated with that. And then kind of like what we did with the UV Vis with like all of our cuvettes with the ammonia assay, as well as the Gatorade assays, when you get a different spike, that's just going to correlate to how much of that thing came through. So we had a bunch of liquid that had like very little in it, very little in it, very little. And all of a sudden, a lot of stuff started passing through the filter all at the same time. And that is just representative of these beeswax-like molecules that we're starting to find. So yeah, we're going to get to play with some data that looks kind of like this in a few more weeks. But you've had a little bit of exposure to it. Cool.
Don't worry, there's more.
There's more.
There's another one.
There's another one.
Now, this is not my slide, but it's pretty much the same thing. So I see Olivia is explaining the methods here.
And this one is GCMS.
Yeah, this was GCMS, like Julie explained. So it was just a way to separate all the different components of this mixture on the body, and to try to figure out, like, to discern what each individual component that made it up is. So as we can see, the resin was vaporized and then passed through a column. It works like a gel. It works just like a gel.
Yeah, kind of.
It's just the same thing.
So the resin being vaporized is a gas chromatography method. So we want to try to vaporize everything and put it into a gaseous form so that we are sampling the gas. We're going to do the same thing with caffeine in a few weeks, where we're going to have to essentially put caffeine and all of its oils into a thing, boil it off so that we have like just a small little bit of liquid left. Take that tiny bit of liquid and then vaporize it once more. Yeah, it's gonna be fun.
Mhm.
But that's just a way to like get purified pieces of this. So we can
Okay.
find all the specific
Notably, this graph is not from actual skin of the body. This one is from the wrappings. So it's kind of showing us the difference in the composition of like the cloth used to wrap up the body versus the oils present on the skin itself. So, and then also this one was from a female. Yeah. Oh, fun fact. the reason why this wasn't super unethical and terrible was because this method only requires a very small amount of sample to produce our data. So instead of going into museums and like needing a crazy amount of like historical artifacts, just like a small sample was taken. So they agreed to provide that. Okay, I think they filled it. There's like a bunch of them. Okay, so what was she going for here?
We're trying to show the different regions as things push through the retention time on the graph.
Okay, so yeah, she's kind of classifying what each little point on the graph indicates. So like the smaller things, these were associated with plant oils or animal fats used as the base of the emulsion, I guess. And then the slightly longer, slightly larger molecules were the beeswax associated molecules, and then the biggest things were these triterpenoid acids, which are also derived from plant material. Because of some writing about the process, like the mummification process, it was believed that there would be like petroleum biomarkers or bitumen as a primary component, but actually we didn't find that at all. It was mostly plant-based oils and the beeswax. So that was kind of cool, I guess. Yeah. And then this is just telling us about our results. So we found that they're basically whatever writings there were about the process of mummification, they are not universal, and they are also different depending on the region in which each body was found, and the available materials there. We learned from the general increase over time of using beeswax versus other oils, like such as like coniferous resins, that it was actually like it was likely discovered by embalmers over time that this method had a better outcome of preserving the body. So it kind of shows us that knowledge over time. Yeah, so basically the thesis of this whole thing is that we really don't know. We don't know that much. We claim to know more than we do. And it's a life lesson.
Awesome. Cool. Anything else to add on? All right, thank you. Cool. Who's our next victims? Cool. Theo, you had your hand up first. You were chemistry poisons.
Ava, you're a third person then.
Oh, I genuinely don't remember. Sorry, I don't think I'll be helpful with you. I'm sorry.
Yeah.
That's fine.
Do you have anything?
Do you want to
Do you want to say?
what if you do the production and the methods of the discussion of the fishing?
That's all right.
Chemistry poisons? Yeah. Take it away.
Okay. So, are we using? Is this more related?
Yeah, I have some figures
Got it.
for
Okay.
later.
So there was this course called Chemistry of Poison, and it's supposed to be an interdisciplinary course that combines well, different mostly organic sciences. It helped show how these different what was it? It was biology, pharmacology, toxicology, and the humanities, how they all connect chemistry too. Uh, problem, is this like the beginning? This is like a really broad overview. That's where I should start.
Yeah, this is just.
That's the, oh, got it. Okay. Um. Okay, so initially the course wasn't meant for people who were like it wasn't supposed to be an upper level chemistry course. It was supposed to like that it wasn't for people who were specifically in chemistry. It could be people who wanted to go into pharmacology or just pre-med students too. But a lot of different kinds of people took the class. So you had 52% biology majors, 22% chemistry majors, 19% engineering, and seven other course or people in seven other majors. And most of those were also STEM. Initially the course had
Those are just figures for later.
Okay.
initially the course had a teacher or the professor was in chemistry and then they had a TA that had a background in pharmacology. And because of a change in the teaching assistant after 2 years, class tried to focus more on the chemistry aspect rather than the more medical side. Organic chemistry 1 and 2 were prerequisites for the class. So students were typically juniors and seniors. And the final project has taken two different forms. One was to write or edit a Wikipedia page on the topic or on a certain topic about poisons, or prepare a poster that can be presented to the public by the end of the semester. For the poster, there was a focus on creativity, something that looks nice, but also something that clearly communicates the idea. And another final project, or part of the final project, was a souvenir. So this is like something that the students would
Yeah, do you want to take us away with some of the?
I think that's it.
Here, can I? There's
I
more
don't
things.
really understand the souvenirs.
So, okay, so for the final project, one of the things that I wanted to,
I'm just like scrolling through just to see things. Okay, cool.
so.
Yeah, like examples.
Yeah, so one of the sort of the main thing of the final project that I wanted to focus on, because this is an interdisciplinary course, is that its focus was on sort of creativity and learning how to present scientific research and findings to the public and learning what to include, what not to include. And that's just like, I just wanted to highlight that skill because that's such an important skill for communicating scientific findings. And it's something that can be very easily left behind in general science courses and just when you just get really stuck into one field. And then the souvenir, which is sort of a physical object that the students were able to, in the beginning of the semester, come up with ideas and vote on to sort of what shape the souvenir will take. And it would include parts of each person's final project. And that was just also a part of just like learning how to sort of summarize your findings and your research and your project into a way that the public could understand, which is also a really important part of science. And it's just also something that can be very easily forgotten about. But when you get, if like, even if you spent a decade researching something and you like found really, really important results, if you can't communicate those to the public or to your peers, those results aren't gonna go anywhere. And so yeah, it's just a very important skill. But another part of this course was there was a survey taken for the class in 2015, 2016, and 2017. Yeah. And
you can be in charge.
okay. And so for this, there was a part that was just why'd they take it. A lot was just for intellectual curiosity, which makes sense. It's a cool course. For some, it was to earn credit for their minor. For some, it was recommended by other students, which increased in the 2016-2017 course as like because students had taken it in 2015 and recommended it. Some for their major, and then some because it was relevant to their career goals. And so, yeah. But and then the 2017 group was also got some, both the 2015 to 2017 group got questions asked. The 2017 group got some more questions asked. And so these are the 2017 groups, and I just wanted to highlight a few of them because I thought they highlighted how beneficial the interdisciplinary parts of this course were. So one was the amount of chemistry involved with historical events, and that just sort of shows the crossover of studying history and also studying chemistry and how there are ways that those interact. Another is how they were surprised that they needed to understand a lot more biology, which is very important in chemistry, but it can be easily forgotten about. And then the last one on this one is just, the ocean is full of chemical weapons. I don't know what that means. I would love to know what that means, but it's a mystery for the ages. So then we have the feedback from the 2015 to 2017. And so there were, again, just a few things I wanted to highlight. One is, this is a little hard to see, but so as someone who's very interested in bio, I'm obligated to say, bring a bit more biological systems into the course. I was always interested in some of the more specific effects/symptoms were for the poisons we talked about. And while it was relatively easy for me to look up, barring the fact that I'm certainly on a Homeland Security watch list because of it, it would still have been nice to hear from the professor. And so this just showed that like there is still a lot of room to grow in these types of courses and a lot of room to be like, oh, we can include more from this field and less from this field, or more from both fields. And the last one on this slide is just, I greatly enjoyed the intersection of anthropological/critical thought via the writing homework and the application of chemistry. I also liked how intricate the mechanisms of the poisons, and that the chemistry was applicable. And again, this just shows that there is so much intersection between science and the humanities. It's not just between scientific fields, it's between all fields. And yeah, that was just a very, very cool part of this course that it showed that. And so these were just some figures on what the students found were most useful, which were the lectures and the workshops. And then we have here, so they all took a survey and rated different statements between 0 and 5, strongly disagree to strongly agree. And then these are the averages. And so some ones I wanted to highlight were just that the students agreed that the study of both chemical and biological principles improved their understanding of the activity of poisons. And they pretty strongly all agreed with that, with an average of 4.45. Whereas the idea that studying them separately would have made learning more efficient was pretty strongly disagreed with an average of 2.24. And then the studying of both science and non-science content at the same time was useful for their learning, and that was also pretty highly agreed with. And again, just highlight, there's so many benefits to studying humanities and science and different scientific fields at the same time that can really just increase everybody's understanding of it. So just some conclusions I just wanted to highlight are just that, like, the increasing complexity and politicization of science means, like, the benefits of interdisciplinary courses have increased just since we've gotten more complex in all these fields, and these fields have gotten further and further apart as we've gone deeper into them. Learning those intersections is so important. And these courses, they can improve student problem solving, they can help them develop new perspectives, and they can show them how to better apply information from one scientific field to another. And they can show how these fields relate to each other, and just how science is just one jumbled mess of topics that can seem separate, but they all relate to each other in some way or another. But also there are challenges to running these courses, how the fields are defined, to what degree, which fields, like these can be very challenging things for a professor to do. And so what that also means that there's a lot of room to grow here, and there's a lot of room for creativity to be brought into these courses and to make this stuff interesting for students. And then just the last point I wanted to say is just chemistry and toxicology, but like chemistry is just, it's such a good field to do these types of courses with, because it's essential science, because it's like every science relates to it. But also because, especially in toxicology, understanding the biological effects of toxins is so important, because if you don't, and you're studying this molecule, you're designing a molecule, and you don't know if it's going to be harmful to the environment, to humans, to other animals, then that can be extremely problematic. And that's happened because they just did not understand, they did not spend the time to study and learn those biological effects. And then there's also just like, there's so much in the world and in just of how everything's interconnected. But yeah, that was the paper. I thought it was really interesting. I don't know if you did, because it's more about course design than poisons, but I like that kind of stuff. Yes.
Where was this course offered? I think I missed that part.
I do not know. The paper did not say.
That's awesome.
Or at least if it did, it was very brief and I could not find it again.
That's really
A lot of college course lists out anyway.
University of Rochester,
Oh, cool.
and also with Lake Erie College of Osteopathic Medicine, whatever. But yeah, Rochester.
I'm not registered, all they're teaching is broom making, it's sweet.
This seemed like a really Hampshire class, and that's what I kind of
Yeah.
loved about this one.
Yeah. Now, the deeper I got into the paper, the more I was like, oh, wait, this is actually
Yeah,
cool.
and then they had like the little pennyroyal, like
Yeah.
like the poison that you can make to make people go unconscious if you want. And please don't do that unethically.
I would ask, for your first, or I guess it's like these past 2 months, do you feel like you've been able to draw interdisciplinary connections in New Hampshire courses? Like, do you feel like your experience here related a lot when you were reading through this, or?
Definitely. I think my experience, like, growing up with two science parents related a lot to this, since I've always been in the, like, oh, everything's interconnected, but like definitely in Hampshire, I've started learning a lot more about like I took animal physiology last year, and I was just like, oh, this all connects. Chemistry is related to animal behavior, biology, like it's all interconnected. And I think so it's just very interesting to see all this stuff. I was like, yeah, it is. And like these courses can be really cool.
One of the, oh, sorry, yes, you,
I was just clapping again.
yeah, we clap again, great,
Fine.
good job, Matt. One of the grants that I want to write for like the next grant cycle, it's early January, but it'd be for the upcoming year, is to try and transform part of our education where essentially you would have each year probably 4 big projects, and there would just be a bunch of classes that are associated with those 4 big projects. So something that might be like animals, animal behavior and something like that. And it's not just animal behavior, but like we could do chemistry on that. We could also do microbiology centric around those animals and things like that. And you have an entire track that kind of dedicates to that. Jenny and I have talked about where we submitted a different thing recently for these blueberries and just like how they might work together in the environment. So having Jenny's classes on like plant and disease ecology, my classes on microbiology. Then you take my biostatistics class, and then Kenneth gets to come in and talk to you about actual statistics. And like that is one big project of multiple courses. This seemed really inspired by like that similar idea. And I just love the idea of like a chemistry approach in poison class.
Yeah.
There's a couple other fun examples that they've got thrown in here. So yeah. Cool. Good job, Bill. You get third clap, third applause.
The opportunity I get to.
We got 2 more. We've got chemistry of smell, and we've also got an ag decoagons. Which one you want to come up?
Oh, you're one.
It's its own animation, apparently.
Period.
I know, spooky. It disappears.
What's on? Sorry. Sorry, my water's all over the floor.
Oh no. It's carpet. And it's warm outside. It'll evaporate.
It's warm outside.
Or it's warm in here. It's warm. We've turned the heat on, so it's warm and dry, so it'll evaporate. That's the wording that I was actually trying to say.
Hello, everyone. How to control a vampire. Yeah. Okay. This information from our presentation is a review on how anticoagulants are being explored as a method to control the common vampire bat population and prevent rabies transmission. There are 3 species of vampire bats, but we're focusing on Desmodus rotundus because it's the primary species responsible for rabies in Latin America. These other 2 are, you can read, and another word, this one is the hairy-legged common vampire, or no, the hairy-legged vampire bat. This one's the common vampire bat, our star of the show. And this one is the white-winged vampire bat. Hey, oh wait, go back one. Sorry, one more thing. The reason that we're focusing on this one is because it primarily feeds on mammals. These other 2 feed on birds. Okay. So, rabies virus or RABV is a major health issue in Latin America. It's transmitted through saliva when it comes in contact with an open wound or like a hole, and it's almost always fatal for both humans and animals once symptoms appear. During the 1970s, rabies outbreaks became more frequent as cattle ranching expanded. That's because the abundance of cattle provided an easy, stable food source for the vampire bats. Before the 1970s, not much was known about the vampire bats' ecology or life history. But once ranching increased, scientists began studying them more seriously, especially since they were shifting to feed mainly on cattle. Humans are more likely to contract rabies from bats than domestic animals, which, like on a global scale, it's usually the dogs and cats that are giving us rabies, but in Latin America, it's bats. Yeah. Okay. They have specialized teeth that makes small but deep wounds that keep bleeding even after the bat is gone. Each vampire bat can consume about 7.5 liters of blood per year, which is a huge amount considering their size. These wounds they cause more than just blood loss. They also can lead to anemia.
weight loss, decreased milk and meat production, and then like other types of infections. Because of the economic and health impacts, people tried many like different methods like nets or protective grids or even explosives to burn their roosts, which is like the caves they lived in. But these methods were not selective and often killed non-vampire bats, which threw off ecological balance, affecting pollination, seed dispersal, and natural pest control. So people don't like bugs more than they don't like bats, I guess, and they decided to be a little more specific when it comes to like control methods, so they're more targeted and less ecologically damaging, which is why we're trying to make anticoagulant.
So this paper was essentially trying to see what happens when you poison these bats in different ways with anticoagulants. And the bats have these grooming rituals. They groom each other and themselves for 2 to 3 hours throughout the day. And so if the anticoagulant is like applied to the back of the bat, then they ingest the poison and it reduces the amount of effective platelets and clotting factors in their blood, and then they bleed to death internally. So it was initially developed as like a rodenticide for rats, and then it eventually became a human anticoagulant that we had. And most of the papers used warfarin, and then all of these other ones were like just a more powerful version of warfarin. And some would use that.
So this paper was like a meta-analysis, so it took information from a bunch of different studies. So this is flowchart is kind of explaining how they got all their studies. So they started with their search terms with the Desmodus rotundus and anticoagulant and got a bunch of papers from there, and then they excluded them they went through and took out papers that were off topic or didn't quite talk about like control of rabies or the interaction with cattle. And then from there, they took out duplicates, they found more papers, and then they took out papers that were like old or incomplete opinion pieces or papers that didn't have like qualitative or quantitative like data on it. And they ended up with 12 total studies they were looking at. So of the 12 studies, they're about evenly split between 3 types of studies. There's laboratory studies, which looks at the bats in like individual chambers. There's ranch or field studies, which are like the subject they're primarily looking at is cattle, like at farms. And there's cave studies where they looked at the bats in their natural habitats and their roosts. And through these 3 types, they were either the anticoagulants were either directly applied or indirectly applied. The direct application meant they were applied directly to the bats, which spread to the grooming rituals that Amalia talked about. And then for the indirect application, they were either the anticoagulant was either added to the cattle and their wounds or to the bat's environment, which had to be like cave walls, stuff like that.
So most of the studies were looking at the variable of scent effectiveness, which was basically the number of living rats at the beginning of the studies compared to the number of living bats at the beginning of the studies and then dead bats at the end of the study. But none of the studies were actually looking at the transmission of rabies. So they all sort of just assumed that like if you kill all the bats, then there's no more rabies. So we're just looking at the statistics about the bats. I still keep saying rats instead of bats. I'm so sorry. There was a 15-day average of length of the study, and there were 1 to 546 bats treated, and then 68% of the studies used the direct application method that they talked about. In most studies, there were 10 to 800 living bats pre-application, and then 1 to 350 living bats post-application. And with indirect treatment, there's 55% to 100% effectiveness, and with direct treatment, there was 32 to 100% effectiveness.
Okay, so one of the main issues that this meta-analysis brought up was that there is a lack of studies after the year 2000. Current rabies control practices in cattle use 20 to 50-year-old references, which doesn't quite make sense. Yes, most of the studies in the analysis were pre-2000. This could be because of tighter animal welfare regulations. So it's like less of a problem because the cows are more protected. And because the studies led to the reduction of bat populations because they were flowing them up. Most of the studies didn't consider in the bat's individual traits like age and sex, which influence their behavior. I'll talk about that in like 2 slides. And the behavior of rabid bats versus healthy bats, and the duration of effects and frequency of anticoagulant application, like how often they apply them, how long they last on bats. And then warfarin is the most widely used active anticoagulant ingredient. Its effectiveness decreased in the 1970s, leading to the creation of super warfarin rodenticides. Those two were kind of earlier pesticides. And the most commonly used method of application was directly to the bats. You can also apply it to the wounds of the cattle, which are the spots the bats are going to target. But only 40% of the bats actually make contact with cattle sufficiently enough for the anticoagulant to get onto them. And then that cattle contact varies due to the cattle's movement, temperature, and posture. So if the cows tend to be less, I keep saying cows, cattle. If they tend to be more nervous, the bats don't want them. Okay, and then bat routines. Adults usually leave their roosts to feed and then groom daily, making them the best targets of anticoagulant poisoning. Juvenile bats are worse targets because they have more rabies virus antibodies and they have less exposure to the anticoagulant poisoning. Thirdly, when males reach maturity, they leave to form new colonies, increasing rabies virus transmission. There are no studies found examining these patterns, so we don't know that much about them leaving to form new colonies. We just, that's just been observed. Yeah. And then anticoagulant vampiricids were shown to decrease bites and increase bat mortality. The absence of bats in caves after treatments could be due to the disturbance during application rather than treatment itself, like it just scared them off. Researchers don't recommend trying to control bats during active rabies virus outbreaks, but advise people to take action at the onset of bat attacks because when they have rabies, they have rabies. Some bats were preyed on by other animals, which leads to the possibility of secondary or indirect anticoagulant poisoning. And then bats also share caves with other bat species, which brings up about the possibility of poisoning harmless bats. And that's not good. That's not our target. So overall, reducing D. rotundus populations may be counterproductive and lead to further spread of rabies virus while negatively impacting other organisms. So it's not really recommended.
Cool. Awesome. Good job, y'all. Great. Cool. Let's do the next group because we got 13 more minutes. We got chemistry. Oh, how do I do this? How do I do this? Because you gave me this in PDF form. Yeah, come on up, y'all. Just trying to figure out the best way to present this. Cool. We got one more paper. We got another review. Smell of death. Take it away.
All right, so, let me start with the postmortem by chemical treatments. And so after death, the body undergoes autolysis, digestion and petrifaction, which is microbial decomposition. These processes release volatile organic compounds. And so autolysis, I don't think I'm pronouncing it right, but it begins within minutes to hours post-mortem, driven by enzymes breaking down cells, and petrifaction involves microbial decomposition, especially in
Anaerobic.
anaerobic bacterial, while producing gases like hydrogen, sulfide, methane, and catamaran.
Nobody cares.
And the review talks a lot about the volatile organic compounds, which are chemicals that evaporate and release vapors. These are gaseous byproducts of various biological processes, including decomposition, and can provide crucial information about the circumstances surrounding the death, such as post-mortem intervals used in forensics. Techniques like the GCMS help identify odor and quantify volatile organic compounds, as does the GCEAD to analyze insect responses to the volatile organic compounds and linking chemical signals to ethological evidence. And these are the gases that are released during decomposition, and it like divides into like early stages and late stages. And I don't think I could pronounce most of those, but those are like the early stages and like the later.
Time, temperature, soil types, pH, and insects are some of the intrinsic factors which impact the profile of a corpse's VOCs, volatile organic compounds. Of these, one of the most important is temperature, even though the only chemical class it was found to affect is non-cyclic hydrocarbons. Studies on the effects of temperature on VOCs indicate that a lower temperature usually leads to fewer VOCs. One study compared summer and winter VOC profiles of pig carcasses and found 116 in winter and 256 in summer VOCs. Now, all the VOCs found in the winter trial were also identified in the summer, meaning colder temperatures also don't seem to produce unique VOCs. It doesn't seem that lower temperature prevents specific VOCs so much as the increased temperature promotes a greater concentration of them, since higher temperature hastens the rate of decomposition process and increases insect activity. Moving on to the effect of soil, which is still on the slide. Sorry. The first impact which any type of soil will have on a buried cadaver is the prevention of VOCs that require oxygen. Less permeable soil like condensed clay can prevent odors from emanating from the body, while the most VOCs detected from the buried body appear when the soil moist and acidic. More VOCs are detected from buried bodies during days with heavy precipitation. And lastly, not only do the VOCs of a corpse in a shallow grave reach the surface more easily, but it seems a body in a shallow grave also creates more VOCs than one that's buried deep because of the effect on microbial activity caused by higher level of oxygen. The effect of water in a study between dry pig carcasses and submerged ones, a total of 74 unique VOCs were identified between the two groups, with 41 in the submerged trial and 70 in the trial one. Submerged pigs showed no evidence of any cyclic hydrocarbons or ethers, I did have butanol was the only aldehyde in the entire study. Might mean that butanol is unique to water. The bodies left in the water for a certain period also had a wide range of alcohols, sulfur compounds. Another thing to consider is how a body's VOC profile will be affected when left in an urban environment. An experiment aiming to study this left pigs and pig corpses in a field, a forest, and an urban site. A total of 35 common VOCs were identified in each of the sites, but there was still a multitude of differences between the profiles of each location. And the most distinctly different profile was that of the corpses in the urban biotope. Bodies in urban environments are inevitably affected by numerous contaminants, which disguise the VOCs with a cadaver, but additionally, urban environments can lead to a somewhat unique combination of the extrinsic factors that wouldn't normally be seen in other open air places. Bodies left in the wake of natural disasters that struck urban environments were also discussed, and the results found are kind of a culmination of the rest of the factors so far, which is bodies buried in rubble have fewer VOCs depending on how thoroughly they're buried, and produce more of the unique VOCs that result from anaerobic processes. And yeah. Intrinsic factors are mostly parts of the body and its cause of death. Different parts of the body, from fat to bone to muscle, or even excretions, produce their own VOCs, and studying these varying profiles on their own is important for situations where there isn't an entire intact corpse to discover. Anaerobic bacteria digesting intestinal tissue produces gases which bloat the corpse. Breakdown of fatty acids produces an abundance of aldehydes. Muscle proteins are more stable and break down more slowly. So those are just some differences between different parts of the body. And different locations can also cause bacteria communities on and in the corpse to evolve differently, which will also lead to variations in VOCs. In addition to individual parts of the body, excretions also have to be considered whether you like it or not. The odor of blood closely resembles that of a corpse, and the VOCs it produces change greatly over time as it decays, which makes it very useful for learning about death without a cadaver to go off of. Even the residual air of a scene can be studied. An analysis of expired air found 63 VOCs, whereas 38 were found in urine and 46 in blood. And the three all exhibit the same kind of chemical diversity. Surfaces that have been in contact with decaying remains are important to consider as well. Glass and metal rapidly emit what scents they have captured, whereas plastic releases a high rate of smells for a longer period of time. An investigation found that blood follows a comparable degradation process no matter what surface it's on, be it cotton or aluminum. So there is a period of a few months where you can tell what surface the blood was sampled from. And then toxicology is also an intrinsic factor relevant to the VOCs emitted by the body, because toxins have their own scent profiles and different impacts on the body, which will be identified from the
Going off of what I said earlier, so for etymology, carrion insects, one of which can be seen down here. This is a carrion beetle, also known as the burrowing beetle. And it is a type of insects or a group of type of insects that are associated with decomposing remains. And they usually appear they appear in succession patterns, which means certain types of carrion insects like show up according to the stages of decomposition of the body. So some show up during the fresh stage of the decomposition, the active stage, or the later stages. And then the use of this is that when if you can identify
the types of insects that are around the body. You can also help that identify the PMI or the time elapsed after the death. After death, yeah. The specific VOCs that are mentioned in this like time or chapter, I guess, was dimethyl disulfide, diethyl trisulfide, phenyl, and idle. Oh, sorry. Carrion insects. The reason why they're there, if they have a variety of reasons to be at the site of the decomposing remains, mostly for finding a mate, food, laying eggs, and offspring development usually. The methods of identifying like insects and like these insects to identify the PMI, the methods are usually EAG or electroantenography. Apologies. And GRASS chromatography, electroantenographic detection. Oh yeah. So EAG is when they input like metal like antenna, if you will, in place of their usual antenna in order to identify when the brain reacts to like the odor of some of these VOCs. And then GCEAD like can measure specifically what odors they're reacting to. EAGs were found in several studies to not be that reliable of a method when identifying one VOC at a time, but in conjunction to like others, like two VOCs, for example, it was more effective. They also found in the study that specifically with the insect, Dermis fresci, that depending on the sex and physiological state, some of these insects, the VOCs act as either a repellent or an attractant for that insect in particular, which is an arthropod or arthropods. So the males of Dermis fresci, usually appear first. The EAG showed that they react to the decomposing remains by the time that they get to the active to later stages of decomposition, but during all stages of decomposition, but they only are willing to go to that body once it gets to the active stage of decomposition. Meanwhile, the female version of that insect will refuse to go to the decomposing site of decomposition unless the males are already there and are spreading pheromones to attract like the women, sorry, the females. So that's the sex, and then for the physiological state, that's only for the females that are whose reproductive organs have like fully matured. So for the younger versions of this insect, they are fully willing to go over to the decomposing like remains around the same time as the males. The insects themselves, so again, very useful for identifying PMI. But other methods are using the insects themselves to identify PMI, the post-mortem interval, or how much time is elapsed. Because they similarly, when they die or when they start decomposing, they also let off the same exact VOCs, so the four listed above. So they can also be used as decomposition for the VOC collection. Similarly, maggots can also be used and are useful in PMI estimation because again, same VOCs that they give off. And this is especially useful when there isn't actually a body for them to sample. There isn't a body, but there is like these guys, little insects. And so they can still identify how much time it lives since the body. Yes. And then there are also flies, which similarly to the last few.
Try and wrap it up because we're at.
Yeah,
time
we
a
are
little bit.
over. I apologize.
You're good.
They also use human remains detection dogs or cadaver dogs, and they train dogs. It can be a variety of breeds of dogs. Specifically, they like to use shepherd dogs, but they said that as long as they have a good attitude, it does not matter what type, really. They use dogs because they are highly sensitive and display high selectivity. When they are training these dogs, they are trained from multiple stages of decomposition and on multiple materials so that they can use it and generalize it to real themes when they would be used. When they train these dogs, they usually train on either pseudoscience or like the natural like stuff, so like decomposing matter, or rather they said grave soils. Grave soil or just normal soil. Except there are problems with both of these. The pseudoscience usually lack the VOCs specific to humans. So the accuracy when training the dogs is off, which means applied to like real world field, it's not going to be quite as good. And then the availability and legality of using natural like remains is disputed, and it's again hard to obtain, and you have to go through like legal loops and jumps and whatnot to obtain these in order to train the dogs.
I'm gonna cut you here. Sorry.
Sure, that's fine.
But let's give them a round of applause. Thank you for that. Cool. I will see you all next week. We've got Ananda I'll see you tomorrow morning. We're doing struvite. We're gonna make some kidney stones or fertilizer, depending on your context. Same thing.